Glycolysis is the first step within glucose metabolism, and in this process, we see the oxidation of glucose into two pyruvate molecules. And those two pyruvate molecules can further be processed and fed into the TCA cycle to further metabolize the carbons. Now, when we begin glucose metabolism, we start off with a glucose molecule, which is a 6-carbon sugar. It's important to make note that the hydroxyl group on carbon number 1 is facing down, so this is in the alpha position. And this is carbon 1, 2, 3, 4, 5, and then this is carbon number 6. In the first step of glycolysis, we see a phosphorylation event, which is catalyzed by the enzyme hexokinase. So hexokinase, first of all, it's hexo because we're dealing with 6 carbons, and it's a kinase because kinase is involved in phosphorylation events where we're dealing with ATP or ADP. We also have magnesium 2+, which is simply just there to balance out any charges and help the reaction happen, because since we're dealing with phosphates have a negative charge, and so we need a positive charge to help stabilize the reaction. So we use ATP, we hydrolyze the terminal phosphate on ATP, and we add it on to the 6th carbon of glucose. And as a result, we get glucose 6-phosphate. Now, glucose 6-phosphate, it's still a 6-carbon sugar, but there's a phosphorylation on carbon number 6. So now there's 2 reasons why we want to phosphorylate glucose. The first reason is that when we phosphorylate a sugar, we're marking it down for degradation. All right, so when we mark something with phosphate, we know that, okay, the cell has to break it down. The number 2 reason that we mark glucose with phosphate is because we know that the cell membrane is permeable to certain types of molecules. So that's specifically molecules that are uncharged. So originally glucose is an uncharged molecule, so it can readily diffuse through the cell membrane. But when we add the phosphate, so if we draw the structure of phosphate, it looks something like this. So when we add the phosphate onto glucose, now glucose is negatively charged because of the phosphate, and it won't be able to leave the membrane because charged molecules cannot leave the membrane. So those are the 2 reasons why we are phosphorylating glucose. Then we move on to an isomerization event. And the only reason we're going to isomerize is because we're trying to create a more of a symmetrical molecule so that when we break this sugar in half, we get 2 symmetric products. So we use a phosphohexose isomerase enzyme. So it's an isomerase because we're simply going to switch from an aldose into a ketose. So we use an isomerase. And then it's hexose because it's we're dealing with 6 carbon sugars, and it's phospho because we phosphorylated. We are once again using magnesium 2+ to stabilize any charges. And then we get fructose 6-phosphate. Now, fructose 6-phosphate, we can see that now over here on carbon number 1, we have a hydroxyl group, and we can phosphorylate that hydroxyl group to get a symmetric molecule. Then on carbon number 1 and carbon number 6, we'll have phosphorylation events, and then we can break this molecule in half. So now we take fructose 6-phosphate and we convert it into fructose 1,6-bisphosphate. So essentially we're utilizing ATP, we're hydrolyzing it, and we're transferring that terminal phosphate onto carbon number 1. And we're utilizing phosphofructokinase 1, which is a very important enzyme in glycolysis. And then we have magnesium 2+ to stabilize any charges. Now we can see the name of this product is fructose 1,6-bisphosphate. Now, fructose just refers to the sugar. 1,6 is the location of where the phosphates are, and it's phosphate because we phosphorylated, and bis simply means that the phosphates are not right next to each other. They're in different locations. So now the next event is breaking fructose 1,6-bisphosphate in half, and we do that through the enzyme aldolase. So aldolase will break fructose 1,6-bisphosphate into two products, and those two products are going to be dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So those are our two products. Now, if we take a look at the structure of both of those products, they're symmetric. So we've got a phosphate on this one over here. We got a phosphate on this one over here. And then both of them have 3 carbons. Now the difference is where the hydroxyl and carbonyl are located on both of these molecules. In dihydroxyacetone phosphate, we see the carbonyl on carbon number 2. We see the hydroxyl on carbon number 1. But if we take a look at glyceraldehyde 3-phosphate, we see the carbonyl on carbon number 1 and the hydroxyl on carbon number 2. And that's the difference between the two molecules. Now, we want, when we're dealing with glycolysis, we only want the G3P. Okay, we're not interested in the dihydroxyacetone phosphate or the DHAP. So what we do is we convert the DHAP into G3P. And we do that through an enzyme known as triose phosphate isomerase. Now, triose phosphate isomerase, it's a triose because we're dealing with 3 carbons. It's a phosphate because they're both phosphorylated. And it's an isomerase because we are simply converting from one form to another form. So we convert that dihydroxyacetone phosphate into G3P. And now we have to make note that every step from now on forward, we're going to be dealing with 2 G3Ps. All right. So the first step, so up until this point, so steps 1 to 5, steps 1 to 5 are preparatory steps. So in which we're just preparing the molecule to produce that energy that we're interested in. Now we're dealing with the payoff phase. And payoff phase starts off with step number 6. And step number 6 is a very important step because step 6 is coupled to step 7. So we take NAD plus and we reduce it to NADH and H plus. And this transfer of energy this transfer of electrons allows for the phosphorylation of G3P. And we do that through the enzyme known as glyceraldehyde 3-phosphate dehydrogenase. Whenever we see the name dehydrogenase, we can just think that, okay, we have a dehydrogenase, so we must be having a redox reaction. So when we reduce NAD plus to NADH and we phosphorylate, we can see right over here that now we are going to be forming a carboxylate. Over here we had a carbonyl, but now we're going to have a carboxylate because we removed that hydrogen. We removed the hydrogen that was over here and we added a phosphate instead. Now we have 1,3-bisphosphoglycerate. Once again, 1 and 3 just represent where the phosphates are located. And then bis just represents that the phosphates are not right next to one another. They're at different locations. And we can see that we have ATE. Whenever we have 8 at the end, that means we're going to be dealing with an acid. So because this part essentially is the acid portion, the carboxylate portion. So essentially, whenever we see this, we're going to be dealing with an 8, also known as an acid. All right. So the next step is step 7, and step 6 and 7 are linked to one another. So essentially, step 7 is a highly exergonic step, and step 6 is an endergonic step. So step 7 drives step 6 forward. And in step 7, we see the first substrate phosphorylation event. When we talk about substrate phosphorylation, we are dealing with the creation of ATP. And since we're dealing with a transfer of phosphate, we are going to have a kinase. So it's going to be phosphoglycerate kinase, and we have magnesium 2+ to stabilize any charges, negative charges. So essentially, this phosphate right over here, it is transferred to ADP, and we get ATP as a result. Now we're left with 3-phosphoglycerate. Now let's take a moment to appreciate what just happened. We started off with a carbonyl, but it's important to remember that in the beginning, we had a hydroxyl. So we went from a hydroxyl to a carbonyl to a carboxylate. All right. So now if we go back down here, we can notice that this carboxylate has a negative charge. And this negative charge is obviously going to be resonance stabilized because of this double-bounded oxygen right over here. Now, if we take a look down over here, we go from 3-phosphoglycerate into 2-phosphoglycerate. So essentially, we're just undergoing a isomerization event. But when we talk about isomerase enzymes, we can be specific. Mutase is a specific kind of isomerase, and it's involved in transferring a functional group. So we are going to take the phosphate on carbon number 3, and we are going to transfer it to carbon number 2 because we have a mutase. And then we also have magnesium 2+ to stabilize any charges. Now that we have 2-phosphoglycerate, the phosphate is now on carbon number 2 and the hydroxyl is now on carbon number 3, we undergo a dehydration event, in which dehydration, essentially we remove water. And we do that through the enzyme known as enolase. So over here in step number 9, we can see that we have converted an enolate form of pyruvate, and it's called phosphoenol pyruvate. Now, phosphoenol pyruvate, essentially when we lose water, we lose the OH and the H on this carbon over here. So we form a double bond right over here, right? Now that we've formed this high energy molecule in a way, it is going to now, this pyruvate kinase enzyme is now going to phosphorylate the final event of glycolysis, in which we'll see the production of pyruvate. Now, essentially what happens is that this phosphate can now be transferred onto ADP to form ATP. This reaction is catalyzed by the enzyme pyruvate kinase, and then once again, now we have actually 2 cofactors, magnesium 2+ and K+, to balance out and stabilize any charges, and we get the final molecule of pyruvate. So essentially we can see over here that we had a double bond over here, but over here we can see that pyruvate has that extra hydrogen. It has a double bond right over here with oxygen, and we see the carboxylate group at the end. So essentially pyruvate is an acid because it has that carboxylate. Now, it's important to make note actually that we have 2 pyruvates, because remember in step number 5 we converted and we got 2 G3Ps. So we end up with 2 pyruvates, and those 2 pyruvates are going to be further processed through glucose metabolism.